All right, I'll take that as my cue to kick this off. Welcome to the ARO seminar series centered around the topic of the auditory cortex and thalamus. Today we are delighted to have Dr. Ross Williamson from the University of Pittsburgh as our speaker. My name is Kishore Kuchibhotla, and I'll be a moderator for today's webinar. Before starting and during the presentation, if you have any questions, please try to use the chat function and direct it directly to me as the moderator to avoid interrupting Ross during his presentation. Following the presentation, in addition to using the chat function, feel free to raise your hand to unmute and directly ask your question. And any questions that are not addressed during the session, I'm sure Ross would be happy to try to answer them by email if you reach out to him. And closed captioning is available for your use, and the session is being recorded and will be made available for additional viewing. So a little bit of background about Ross. He grew up in Edinburgh, where he studied computer science and neuroinformatics as an undergraduate. In 2007, he moved south of the border, and in 2012, he received his PhD from the University College London, where he worked at the Gatsby Computational Neuroscience Unit with professors Manish Sahani and Jennifer Linden, and his love of auditory neuroscience, I think, really started then. He then worked as a postdoctoral research fellow with Professor Dan Polly at the Eaton Peabody Laboratories in the Department of Otolaryngology at Harvard Medical School. He's now at the University of Pittsburgh, where he has done a fantastic job over the past few years getting his lab to the cutting edge of auditory neuroscience. He's received prestigious fellowships, including the Klingenstein Foundation Fellowship, as well as multiple NIH-funded grants. One of the things about Ross that strikes me is that he is taking on a fundamental challenge in systems neuroscience right now, which is really related to what is the role of different excitatory subtypes and cell types in enabling perception and behavior. This requires painstaking experimental paradigms, cutting-edge optical tools, and also high-end computational approaches to try to understand what this big data actually means. And so in many ways, Ross is that fearless combination of a computational neuroscientist who is now a standout experimentalist. And so without further ado, I am very happy to hand this over to Ross, who will tell us about his latest work, which is related to the brain-wide neural circuits for sensory-guided behavior.
Awesome. Thank you very much, Kishore. And thank you everyone for joining. Can everyone see my screen okay? Looks good? Perfect. I'll take that as yes. Yeah, so thank you again for joining. This is an exciting time for me. I started my lab just before COVID, and it's taken a few years, but everything that we kind of started back in 2019, 2020 is kind of starting to really come to fruition. We're starting to get our first papers out. And as a result of that, near everything that I'm going to be talking about today is either under review or is going to be submitted very, very soon. So constructive criticism is welcome. I would love to know what you guys all think. I'm going to talk very, very generally about brain-wide neural circuits for sensory-guided behavior. And I always really like to start with this kind of silly example. This is a fish show. This is arguably one of the best sensory environments in the world. And I like to show this to make the point that sensory-guided behaviors are really ubiquitous. So auditory information, indeed all sensory information, is almost always associated with distinct behavioral outcomes. So in an example like this, if Trey Anastasio busts into an awesome guitar solo, you're going to turn your head and you're going to focus your attention on him. If they play a song that they've not played in the last 15 years, it's going to elicit an emotional reaction. And although this is a somewhat trivial example, it's a nice example just to make the point that the auditory system is not a hub of emotional processing, nor is it a hub of motor action, nor arguably is it a hub of decision making. So to really understand how sensory-guided behavior manifests in neural circuits, we really need to understand how auditory information flows through brain-wide circuits. So for this ARO audience, this is going to be a little bit simplistic. I like to start right down at the level of the cochlea. So this is a simple schematic of the ascending auditory system that we all know and love. Sound is transduced at the level of the cochlea, and it's then sent through a number of different synapses in the brainstem, the midbrain, the thalamus, before it finally reaches the peaks of the primary auditory cortex. And what's really amazing about the auditory system is that neurons in some of these subcortical auditory nuclei are equipped with the biophysical, the synaptic specializations that support a high fidelity encoding of the acoustic waveform. And on the right-hand side, this is a great example of this. This is from one of Phil Joris's earlier papers. This is an amplitude modulated pure tone, and then this is the representative firing pattern coming from an auditory nerve fiber. And I hope you can see that the the individual amplitude fluctuations in the pure tone are being represented really, really nicely in the neural firing patterns. So the temporal fine structure is getting locked to very, very accurately. As soon as we jump up to the level of the midbrain, this is the beautiful inferior colliculus, we can still find neurons that are temporally precise. This is an example on the right of a neuron that's capable of phase locking almost up to 300 hertz. But this really isn't the case at the level of the primary auditory cortex. So cortical sound representations are purely rate-based abstractions of the source signal. You can see here on the right-hand side that the locking to the actual amplitude fluctuations in this stimulus, they completely go away when we're at the level of the auditory cortex. And one of the reasons for this is that signals can be colored. They can be modulated at numerous stations as we head towards the auditory cortex. And this is something that I just want to focus on for the next few slides, to describe how cortical sound responses can be modulated by a number of different factors. I'm a bit of a literature nerd, and I kind of like to throw out a few philosophy quotes, because this is really something that philosophers have been thinking about for millennia. And this is a nice quote from Aristotle: "Persons do not perceive what is brought before their eyes, if they are at the time deep in thought or in a fright, or listening to some loud noise." And this is Aristotle kind of highlighting the ability of other sensory modalities to modulate a sensory percept. Jumping forward slightly to Descartes, the soul can prevent itself from hearing a slight noise or feeling a slight pain by attending very closely to some other thing. And this is really what I want to focus on for the next couple of slides. And this is the idea that cognitive factors, choose your own buzzword, attention, arousal, behavioral state, internal state, all of these different buzzwords are capable of modulating the way that we perceive the sensory environment. So one way that we can modulate the sensory environment is via acoustic context. On the left-hand side here, this is another kind of classic example from Tony Zader's work back in 2005. And this is a classic example of forward suppression. This is in the rat auditory cortex. And here what's being illustrated is that the responses to the second tone in a pair, in this case, if the interval is less than about 128 milliseconds, are completely suppressed. So as two tones become closer to each other in time, the response to the second tone is masked by that of the first. You can extend that to kind of multiple dimensions on the right-hand side here. This is some Vatsin Sadagopan's earlier work with Xiaoxing Wang. And this is kind of the two-dimensional combination sensitivity map of an auditory cortical neuron. And the experiment here is that you find the tone that elicits the highest response. You yoke that tone, and then you take a second tone and you rove it in relative onset time and relative frequency. And this allows you to come up with these kind of two-dimensional maps showing how tones interact with one another. And in this particular case, the presentation of that second tone really, really likes it, or rather the firing patterns increase when that particular tone is preceded by another tone about 75 milliseconds in the past and about a quarter octave down in frequency. And of course, we can move away from acoustic factors and move on to cognitive factors. This is one of my favorite papers from the last 20 or so years. This is some brilliant work from Jonathan Fritz and Shihab Sharma, looking at how spectrotemporal receptive fields can be modulated when ferrets are engaged in behaviors. And I'm not going to go through all the details here, other than to say that this is a ferret that's carrying out a simple tone detection task. You can estimate receptive fields prior to the animal's engagement in the task. So if we just take a look at panel A on the left-hand side there, we can see a spectrotemporal receptive field with a lovely excitatory hotspot at around about 8 kilohertz. When the animal is then tasked with detecting a tone at around about 6 kilohertz, you can see that when you re-estimate the STRF, you get this excitatory hotspot that occurs, that becomes present at the behaviorally relevant tone. And you can see this in a number of different ways on the right-hand side. So this is a spectrotemporal receptive field that's actually being modulated by the animal's engagement in the task. And more recently, people have got really, really interested in looking at pupil diameter. Pupil diameter is a wonderful measure of arousal. And Matt McGinley back in 2015 really nicely showed that pupil diameter correlates very, very highly with auditory cortical membrane potentials. And this is something that I'm going to focus on a little bit later in the talk to show you some of our own work looking at how pupil diameter modulates cortical responses. But just to return to this example of a fish show, this is what the auditory cortex is doing. So there are almost 1,000 neurons in this video. And one of the main things that I want to convey here is that not one of these neurons is faithfully encoding the acoustic waveform. So really, to disentangle this mess of activity, my kind of neuroscientific worldview, if you will, is that I think it's crucial that we understand the identity of the cells that we record. We need to understand what these cells are. We need to understand their upstream and downstream connectivity. We need to understand the kind of information that they can process so that we can actually eventually understand how we can perceive complex environments, such as this wonderful, wonderful fish show. So that's a somewhat long-winded way of saying that the key focus of my lab is really trying to understand how the auditory cortex is hooked up to the rest of the brain and how this connectivity leads to purposeful behavior and active listening. So I'm going to start with a little bit of anatomical work, with the disclaimer that I am not an anatomist. I find a lot of beauty in anatomy, so apologies if this doesn't meet anatomical standards. This is the excitatory diversity that is present within the auditory cortex. I also want to make the point here that this is also ubiquitous in the neocortex. This is not auditory specific. You find the same amount of excitatory diversity in other sensory systems, but everything I'm going to talk about today is going to be purely rooted in the auditory system. And this is something that I became interested in as a postdoc. I really wanted to try and understand different cell types in the auditory cortex, and I wanted to understand where they projected to and where they sent their sensory information. So very, very broadly, you can break down excitatory cell types into three different umbrella terms that I've illustrated here. Intratelencephalic, IT-type neurons. These are neurons that project solely within the telencephalon. So these are neurons, they primarily reside in layer 5A, or at least that's where the bulk of them are located, but they are located in all cortical layers. And these are the neurons that provide your cortical projections, a lot of the corticostriatal projection, corticoamygdalar, essentially anything that resides within the telencephalon. Skipping over the center for a second and moving to the right-hand side, these are corticothalamic neurons, or rather, these are layer 6 corticothalamic neurons. And this is a projection that purely emanates from layer 6 and targets the medial geniculate body of thalamus. The cells that I'm most interested in, these are my favorite cells in the entire brain, are these guys in the center. These are extratelencephalic cells, ET-type cells. Occasionally they're referred to as PT-type cells in the literature. These are neurons that primarily reside within layer 5B. There are these beautiful pyramidal neurons with these large apical dendrites that project all the way up to layer 1. And these are really the only cells that will take you from the neocortex to subcortical stations. So in the auditory system, these are the neurons that will contribute to the cortical collicular pathway. They also project to the thalamus. And indeed, these are also the same cells that many people have studied over the last 30 or 40 years. Dave Rigo, for example, who's studied cortical projections all the way down to the level of the cochlea. These are these ET-type cells. And something that's really, really remarkable about these ET cells is that they have these elaborate collateralization patterns. This is a figure from an earlier postdoc paper that I published with Dan Pauly back in 2019. This is using an intersectional viral strategy where we inject a retrograde Cre recombinase into the midbrain, into the inferior colliculus. And what this does is it expresses Cre recombinase selectively in cortical collicular cells, so that if we then inject a Cre-dependent virus at the level of the auditory cortex, we can drive selective expression of a green fluorescent protein in just the cortical collicular projection. And indeed, what we see when we slice the brain is that you can see the kind of representative pattern of IC labeling on the right-hand side there. So we're definitely labeling the cortical collicular projection. But this approach labels the entire cell. So if these particular cells drop collateral axons into other downstream brain regions, you can pick them up using this kind of approach. And indeed, you see that these cortical collicular cells collateralize into the non-lemniscal parts of the medial genicular body on the top right there, and also collateralize into the tail of the striatum and parts of the lateral amygdala. So this is something that's very, very unique to ET-type neurons. So if we then just kind of survey all of these different cells, the IT, ET, and CT here, you can see that they really provide a means to propagate sensory signals brain-wide in slightly different patterns. And just to drive this home even further, this is a lovely coronal section from an IT type mouse. So this is an example of some of these IT projections. I've deliberately put in a coronal section here that is from very, very rostral in the auditory cortex. So we can't actually see any cell bodies in this particular section. But I hope you can appreciate that the primary auditory cortex here is projecting to the rostral parts of the auditory cortex, somatosensory cortex, posterior parietal, retrosplenial. It's also projecting to the tail of the striatum. You can see the corpus callosum lighting up incredibly here because these axons are actually traversing to the other side of the brain as well. So these projection circuits really can propagate sensory signals brain-wide. Just a very quick one-slider to kind of show some of the stuff that we're thinking about. I'm not going to present any data other than this today. But we really live in this fantastic world of mouse genetics where you can do such incredible things with different cell types in the brain. I like to call this inception with cells. You can essentially use different patterns of recombinase expression to pull out subsets of subsets of subsets of cells. This is an example on the left-hand side that just extends what I've previously shown by one particular subset. So we're using the same trick as last time to selectively express Cre recombinase in the cortical collicular projection. But what we're doing here is we're using a second recombinase in a Cre-dependent manner, retrogradely injected into the thalamus so that we can pull out the subset of cortical collicular cells that also collateralize into the thalamus. And that's what you can see on the right-hand side here. So green is the cortical collicular projection, red is or rather orange, the M. cherries and the flip-dependent virus. So those cells that are orange are cortical collicular that collateralize to the thalamus. There are additional recombinases that you can kind of throw onto this kind of approach. It gets hairy very, very fast, but it gives you a really, really nice way to understand collateralization and to express useful things in particular subtypes. And I'll just finish off talking about the anatomy by focusing a little bit on the input side of things. I'm not going to go too much into the details here because a lot of this stuff is kind of well known in the literature. We use a transsynaptic rabies tracing approach to identify the presynaptic inputs to these different genetically defined cell types. Here I'm just illustrating the approach, or I'm schematizing the approach on the left-hand side here. Essentially, it involves injecting a combination of viruses whose purpose is to turn a selection of Cre-positive cells using the intersectional strategy that I described. You convert these Cre-positive cells into putative starter cells on which a rabies virus can act. So on the right-hand side here, we can see in red, you can see the cortical collicular cells. And the key thing here is that green represents the monosynaptic input to ET cells. And if we just kind of summarize this data all in one slide, here I'm plotting things marching caudally from the level of the visual cortex all the way to the rostral sides of the brain in the level of the orbitofrontal cortex. And the key point I want to make here is that irrespective of what cell type you're looking at, everything receives input from everything else, just in subtly different proportions. But what I find really remarkable about this is that the inputs that these different auditory cortical cells receive is actually the top-down input, rather, the non-auditory input vastly outweighs the input from the ascending auditory system. So all of these different cell types do receive inputs from the thalamus, as you might expect, but these neurons are also integrating information from other sensory cortices, visual cortex, somatosensory cortex, as well as association areas like posterior parietal, motor regions, and all these really, really cool frontal areas. Here I'm illustrating the anterior cingulate and the orbitofrontal cortex. And interestingly enough, if you believe the colostrum is the seat of consciousness, the colostrum also projects to all three of these cell types. So all of these neurons receive input from near everything else. So this really positions a lot of these cell types in a very, very privileged position to integrate information from lots of different sources and project it to all over the brain, brainwide projections. So now I'm going to kind of move on to arguably the more exciting stuff that my lab is interested in, which is really about how we can monitor and manipulate a lot of these different cell types. Although we do a lot of electrophysiology in the lab, much of what I'm going to talk about today is purely going to be optical. And here I'm just illustrating our ability to use two-photon microscopy to image deep layer projection neurons that are located in the deep layers of the cortex. So on the left-hand side here, I'm just schematizing that we can use the same kind of viral approaches, intersectional strategy, transgenic Cree lines, or whatever you want, to selectively express GCAMP, so genetically encoded calcium indicators, in whatever cells you want. This is a lovely video of some layer 6 corticothalamic neurons in the middle here. This is taken about 650 microns below the cortical surface, which is really, really deep. And indeed, we can extract regular tuning functions from a lot of these different projection types. So this is a lot of fun. More recently, we've started playing around with spatial light modulation. I'm not going to go too much into the details here, other than to make the point that you can use a second laser line in a two-photon microscope to simultaneously monitor and manipulate patterns of neural activity. So what you're looking at here is some neurons in layer 2-3. We're using one laser line to image the neurons. So you're watching the GCAMP activity pulsing up and down in each cell. But we're using a second laser line, and we are stimulating 3 specific patterns of neurons, and we're activating them one after the other. So those are those hotspots that you can see. So this is a really cool holographic optogenetics approach, and it gives us the ability to simultaneously monitor and manipulate neural firing. So hopefully there's going to be lots more on that to come in the next few years from the lab. As I kind of alluded to earlier, arousal, or pupil-linked arousal, is something that the field is getting increasingly interested in. And indeed, all of the imaging that we carry out in the lab, all of the behavior that we carry out in the lab, is always done concurrent with pupillometry. And this is just a nice schematic of our kind of typical approach. This is a head-fixed mouse. We are imaging different cell types, either layer 2-3, layer 5 intratelencephalic, layer 5 extratelencephalic, or corticothalamic. These head-fixed mice are passively listening to pure tones, and we are concurrently recording their pupil diameters. And on the right-hand side here, you can see that over the course of a lot of different experimental sessions and a lot of different animals, you can histogram the pupil states, or rather the pupil dilation states that these animals reside in. So this is a nice, somewhat bell-shaped curve. It shows you that, as you might expect, the animals spend a lot of their lives at a kind of mid-arousal level, sitting at around about 0.6. This is a normalized pupil diameter. And then you will get periods of low arousal and high arousal and kind of everything in between. So the first thing we wanted to do was kind of come up with a nice approach to capture this kind of arousal dependent activity. So these are massive data sets. And because I'm a bit of a math nerd, we wanted to start by using a multivariate linear regression model. So the linear model here is denoted at the top, and we're fitting this to understand how each cell in our data set is modulated by sound and or pupil. So what we're doing is explicitly modeling the cell's evoked response on each trial as a function of stimulus identity, baseline neural activity, as well as pupil and pupil squared activities. So the addition of a pupil squared term is so that we can capture quadratic influences on neural activity. In B, you can see that this is just some kind of simulated data to show that a model like this is able to accurately capture both linear and nonlinear arousal motifs. So you can see here that with simulated data, you can capture increasing motifs, decreasing motifs. These are kind of monotonic changes in how pupil modulates neural activity, as well as kind of regular quadratic functions. It's also quadratic, but we've called it inverse U here for the sake of consistency with the literature. And on the right-hand side there in C, you can see that stimulus coefficients that you extract from this model. So there are separate beta values for every stimulus that you present. These align very, very nicely with the tuning curves of real data. So this model does a reasonable job here. And there are a few nice things that we can kind of grab from a model like this. We can essentially look at the beta values, so the different beta weights in this equation, to give us an indication of the proportion of neurons that are modulated by sound and or pupil. And that's being illustrated in D on the bottom left-hand side here. Here, what we're doing is we're plotting the pupil coefficient, beta p, the pupil squared coefficient, beta p squared, which tells you the kind of relative linear nonlinear proportion, or contribution rather, and max beta s, which is the kind of maximum stimulus coefficient. And from a sound responsive standpoint, this is actually nice because it allows us to threshold the beta S values so that we can get an indication of the proportionality of the population that is responsive to these different pure tones. And on the right-hand side here, you can see how that kind of plays out. Up in layer 2-3, near 50% of these neurons are responsive to pure tones. This decreases as you get slightly deeper, with ET neurons being quite broadly responsive to tones, and layer 5 IT and layer 6 CT slightly less so. Just to further hammer this home, this is kind of just a bit of a data dump showing all of these different responses, as well as showing whether or not we consider them responsive or non-responsive compared to the beta S values. However, what I really want to kind of focus on for the next couple of slides is that averaging in these kind of situations really does throw the baby out with the bathwater, for lack of a better term. So if we average together responses at best frequency as a function of pupil state, which I'm doing here, everything turns out to be approximately flat. This is relatively uninteresting. And one of the reasons that it comes out as flat is because there's an incredible amount of heterogeneity within the data set. There is no reason to believe that the effect of pupil diameter, the effect of arousal on individual cell types or individual cells is going to be homogeneous across a population. So what we wanted to do was come up with a nice unbiased approach to characterize the variability that's present in our data. So we turned to a little bit of unsupervised clustering. Here we're using a method called hierarchical clustering. And what we're essentially doing is taking a matrix of cells as a function of pupil. This is a big matrix where for each cell, we kind of have your pupil tuning function for response at best frequency, so to speak. And we're then running this algorithm which clusters based on Euclidean distance. We can go into the details later if anyone's interested. But the upshot is if you use this kind of unsupervised clustering method, what you end up getting are clusters that represent these kinds of motifs that I described on the previous slide. So here in the bottom, I'm showing you clustered evoked activity. So these are the sound responsive cells, and we're actually clustering on the evoked part of the response. And you can see that there are some clusters that represent increasing changes. So these monotonically increasing cells, monotonically decreasing pupil modulation motifs. And we can also find a number of other neurons that are modulated either in an inverse U way or a kind of regular quadratic. We can further break this down across different cell types. And indeed, we kind of find a diversity of these pupil-related motifs at every cell type, but in slightly different proportions. One thing that we can see across different cell types is that linear, so increasing, monotonally increasing, monotonally decreasing arousal modulations were more prevalent than nonlinear modulations for all cell types. But essentially, we see the same kind of motifs appear again and again in all different cell types in just slightly different proportions. We can then go on and kind of look at how arousal differentially shapes kind of spectral sensitivities, tuning sensitivities. And here we're kind of characterizing the type of linear transformation from low and high pupil states to assess changes in tuning width. So we're using a linear regression approach here, where we're regressing the high pupil responses against the low pupil responses, and we're quantifying the slopes and the intercepts of that regression to get a measure of whether or not your tuning function is multiplicatively and divisibly modulated or additively subtractively modulated. So on the right hand side, you can kind of see an example of this. These are two tuning functions, one in a low pupil state, one in a high pupil state. And I hope you can see that in the high pupil state, your tuning gets broader. You see increases at the tails, and then everything kind of shifts up as well. So indeed, if you then regress high versus low, you see a slope greater than 1, and you see a positive y-intercept. So that basically tells us that this neuron is being additively and multiplicatively changed, modulated. And what we can do is then plot all of these different slope and intercept values, which I'm plotting in C. There's definitely a lot of variability. However, when you run the statistics, it turns out that the vast majority of the modulation, or at least the most modulation, occurred in layer 5 ET cells. So layer 5 ET cells seem to be more multiplicative and additive than most other cell types, which indicates that they are more selective to being enhanced and broadened. during periods of heightened arousal. Okay, so now we're going to jump onto kind of behavior to try and understand what these different cell types are actually contributing to with respect to purposeful behavior. And one of the behaviors that we like to use in my lab is a head-fixed choice behavior, where an animal has to characterize whether or not a sinusoidally amplitude modulated noise is either fast or slow. And that's kind of schematized in the top here. So this is a kind of two alternative force choice task where we're varying the rate of a sinusoidally amplitude modulated noise burst, and then the animal has to either lick left or right, depending on whether the stimulus presented is slow or fast. And when we start training the animals on this task, we ignore all the kind of intermediate stimuli and we just train them on the easy stimuli. So this is more akin to a discrimination task, the slowest versus the fastest. And this is what I'm calling the operant period of learning here in the top right. And we can see that animals can get very good at this task. Once they get to about 85% correct, we introduce all of the other stimuli, and this turns it into more of a categorization task. So the animal is hearing this auditory stimuli, and the animal that has to then label that auditory stimuli with a kind of perceptual label. Is the rate of this amplitude modulation fast or is it slow? And I hope you can appreciate from the psychometric curves on the bottom that once we introduce all of the intermediate stimuli, the animals continue to get better at categorizing these stimuli. And this is best indicated by the increase in slope that you can see in the psychometric curves on the bottom there. We were also very interested to try and understand whether or not the cortex was actually necessary for this task. This is something that has kind of come up in the literature again and again. What kind of stimuli is cortex actually necessary for? So to address this, we constitutively expressed a really potent blue light inhibitor called GTACR2 in the auditory cortex bilaterally. And then we stimulated the auditory cortex on 20% of trials across each day. On the right-hand side here, you can see some electrophysiological control experiments. This is just to convince you that, one, on the left-hand side, we can express GTACR2 across the entire cortical lamina. And on the right-hand side, you can see some different examples, basically showing that we can get near complete suppression in almost every cell that we've recorded from. So on the top right, you can see an optogenetic modulation index where -1 indicates complete suppression. So in response to SAM noise, you can see that everything is near completely suppressed, and indeed the spontaneous activity is also completely suppressed. So this is an approach that works. If we then look at the behavioral results, the main thing that we observe is that when we silence the auditory cortex, performance goes almost down to the level of chance. So performance goes to chance. Other psychometric variables like lapse rates go up, so the animals make increased mistakes on the easy stimuli. You can see that the slope of the psychometric curve also vastly decreases under optogenetic stimulation, and you can see these in the kind of average and individual psychometric curves on the bottom there. So what's actually happening to the behavior when you stimulate cortex or when you inhibit cortex? It turns out that the main effect of inhibition seems to be through altering what I like to refer to as a kind of perceptual choice bias. So here what I'm doing is we're computing a choice bias as the asymmetry index, the difference between left and right choices normalized by the sum, and then we're kind of plotting that, averaging it across a rolling window, and then plotting that as a function of learning. On the top, you can see the light off trials. So this is when cortex is not being inhibited. And you can see that this choice bias, it's centered around 0 for each animal. And it kind of fluctuates very, very slightly. So there isn't too much choice bias going on. But under optogenetic stimulation, you can see that this choice bias fluctuates an awful lot. So what seems to happen is that animals are forced into these bias regimes over the course of several days, where they will particularly prefer one individual lick spout of the two. And you can see that in two exemplar psychometric curves on the right-hand side, session 24 to session 30. In session 24, this particular animal really, really liked the left lick spout, and then in session 30, switched the bias to the other side. And there's some other interesting things that we can kind of look at with this kind of data set. One thing that I really like to look at is how task history affects your current judgments. There's quite a rich history in the psychophysics literature showing that as tasks get harder, you rely more on your prior judgments to guide your present judgments. And indeed, we see this in the control case on the left-hand side. So on the left-hand side here, I'm plotting the probability of a repeated choice as a function of the distance from the category boundary. So as you get further away from the category boundary, the task gets easier. It's easier to categorize those stimuli. Whereas as you get closer to the category boundary, this is far, far harder. And indeed, what you see here is that when you get closer to the category boundary, the probability of a repeated choice increases. So when the task is hard, you use more of your prior choices to guide your current judgments. If we silence the auditory cortex, that completely goes away and everything turns out to be approximately flat. So the animal just finds this task incredibly difficult. And I'll just finish this behavioral part by focusing on the point that auditory cortex silencing doesn't affect sensory motor reports. So the kind of conversion of sensation to action, so to speak, is still occurring. If we look at the no-go rate on the left-hand side here, this is the rate at which mice fail to lick within the response window. This doesn't significantly change. If we look at reaction times, these are response latencies on the right-hand side. This remains approximately the same across conditions. I've deliberately not quantified anything here, purely because I just want to make the point that they are licking. The animals hear the sounds, the animals lick in response to the sounds, but when the auditory cortex is inhibited, they just make their own decision. It doesn't impact the sensory motor report. Okay, so what are cells doing? We recorded from a lot of different excitatory cell types across learning while animals are engaged in this task. And the first thing that I want to focus on is this nice little analysis of layer 5 ET neurons that were significantly responsive to sound during behavior. So in A, first I want to make the point that when animals are engaged in tasks, the proportion of sound responsive neurons goes up. So here we're just comparing across 3 different cell types, layer 2, 3, layer 5, IT and ET, active versus passive. So that's kind of across the board. Active engagement increases the proportion of neurons that are responsive to sound. But what I want to draw your attention to is what I've illustrated with this arrow in B, which is that if we compare early versus late in learning, the proportion of neurons that are responsive to 4 stimuli late in learning in just the layer 5 ET population increases. I'm kind of illustrating this on the bottom there in C, where you can see the actual neural tuning functions as a function of SAM rate. And you can see that in the late case, you kind of get this block pattern occurring. So these are neurons that are becoming increasingly selective to 4 stimuli. And what is so interesting about 4 stimuli? Well, it's 4 stimuli that make up the actual categories within the behavioral task. You have 4 stimuli that are low and 4 stimuli that are high. And indeed, if we start looking at these neural firing patterns in expert animals, this is kind of an example of some of the heterogeneity that you see. In the bottom, you can just see 2 somewhat broadly tuned neurons. Bottom left, this is an ET neuron that is responsive to almost all stimuli. Bottom right, it's a neuron that is largely suppressed by all stimuli. But on the top, we get these really, really cool neurons that are responsive to what I would call the discrete perceptual categories. So in the top left, it's a neuron that is only responsive to the slow stimuli. And in the top right, this is a neuron that is only responding to the fast stimuli. We can quantify this in a somewhat simplistic way, just using another asymmetry index. So this is an index where we take the fast response, subtract off the slow response, and normalize by the sum. So this gives us a value between -1 and 1, which tells us well, it just tells you how selectivity you are to the individual categories. So in the bottom, there are just a couple of examples there. The CSI is close-ish to 0, and you can see that there isn't really any particular category preference. But in the top left and the top right, you kind of got -9 and 0.8. -9 is a slow preferring neuron, 0.8, this is a fast preferring neuron. So what we can then do is take all of these selectivity indexes and plot them as distributions early in learning versus late in learning. And really the main take home, if you take anything away from this talk, I want you to take away this result with respect to ET neurons. If we look at the asymmetry indexes in layer 2, 3 neurons on the left and layer 5 IT neurons in the middle, things do not significantly change across learning. But what you can see when you look at the ET neurons is that you have a somewhat broad distribution early in learning, but this becomes very, very bimodal late in learning. This is indicative of learning sculpting this categorical selectivity that I've just shared with you. What is also really interesting, that this is something that only happens when animals are actively engaged in the task. What I'm showing on the bottom here, that's kind of grayed out slightly, this is a passive condition where we remove the lick spouts from the animals and we present the exact same stimuli to the exact same cells in the exact same behavioral sessions. And you can see that the effect completely goes away. So this is an effect that is sculpted across learning and it is dependent on task engagement. And indeed, this categorical selectivity is also nicely correlated with task performance. On the right-hand side here, you can see that as the performance of the animal, the percent correct, the hit rate, as that increases, the magnitude of the absolute value of the CSI also increases. I'll just quickly skip through this part. We essentially we were concerned that what we might be looking at were some population level effect, because the initial way that we carried out these experiments, we were not matching cells across learning. So Nathan Schneider, a graduate student in my lab, he somewhat heroically redid every single experiment, recording from every single cell type across the duration of learning, longitudinally imaging the same cells across the entire duration of learning. And this is kind of illustrated here. This is one particular field of view of ET neurons that we've returned to again and again across the duration of learning. And there's a couple of nice examples here at the bottom. Neuron 1 here is a neuron that's changing its response properties dynamically across learning to encode these slow stimulus categories and fast stimulus categories on the bottom. And if we then plot the entire data set, here you can see matched neurons plotted as a function of learning. These are the different CSI values. I hope you can appreciate the kind of development across learning of kind of a big blue cluster in the top and a big red cluster in the bottom. So individual layer 5 ET neurons are actually changing their response properties dynamically as a function of learning. And as we kind of saw before, this level of cell activity is not present in individual layer 2, 3 or layer 5 IT neurons. Okay, I want to shift gears very, very briefly just to touch base on the potential for this result to be confounded with what the literature likes to call premotor activity. So we know from prior work, this is actually some work that I was involved in as a postdoc. This is Cameron Clayton's work in Dan Polly's lab. We know from this work that auditory cortical activity can be modulated by motor activity. So in this particular case, licking. So what we're plotting here is electrophysiological data spanning all the different layers in cortex. Animals are not listening to any stimuli in this particular case, they are just licking. And what we can do is then plot things relative to lick onset, and you can see that across all cortical layers, although it's far more prevalent in the deeper layers, motor activity, licking activity actually modulates firing patterns in both regular spiking neurons and fast spiking neurons. We've essentially replicated this using two-photon microscopy in layer 5 ET neurons. So this is essentially showing exactly the same thing that I just showed you on the previous slide, that if you just get head-fixed mice to passively lick, you can see that premotor activity is actually shaping the it's modulating neural firing patterns. Without spending too much time on the methods, one thing that we really wanted to do was double check or convince ourselves that this kind of premotor activity wasn't actually leading to the results that I've described to you. So we developed a generalized linear model-based approach to separate out stimulus and motor contributions to neural activity. And I'm just summarizing this very, very briefly on this slide. So on the left are a couple of beta weight distributions for what I'll refer to as a stimulus neuron on the top and a motor neuron at the bottom. So this is a generalized linear model where we're estimating weights that correspond to different stimuli, as well as whether the animal was licking left or licking right. So when I call this neuron on the top a stimulus neuron, it's because the weights that are being attributed to the motor activity are relatively small in comparison to the stimulus related activity, whereas in the bottom, you can see that motor activity, in this particular case, left lick, is really, really modulating this neuron's activity far more than the stimulus is. But the whole point of this is that this provides us with a statistically rigorous way to identify motory type neurons or putative premotor neurons and remove them from the analysis. That's what we've done on the right-hand side. This is just, I'm throwing the CSI distributions at you again, but this time these are the CSI distributions without premotor activity. So if we take out these putative premotor neurons, we still see the same effect. So this categorical selectivity that I've described to you does not seem to be as a result of premotor or motor contributions within the auditory cortex. And I'll just finish with just one final little result. One of the nice little wrinkles in this task that I've not touched on yet is that we actually present a stimulus directly at the category boundary. So this is a stimulus that is fundamentally uncategorizable, but yet we reward it probabilistically. So we present it to the animal and we still ask the animal to make a choice. What's nice about this is that it actually gives us a very, very pure way to analyze choice, because you can look at left trials, right trials, or fast trials, slow trials, in response to the exact same stimulus. And what I'm showing here is just a little bit of dimensionality reduction. We're taking a population of layer 5 ET neurons. We're using principal component analysis to look at this in a three-dimensional subspace. And this is in response to the exact same stimuli, whether the animal reports it left or right, the population trajectories diverge and go in very, very different directions. So dependent on what the animal actually reports, what the animal thought the identity of the stimulus was, is shaping the neural activity. And you can see this on the right. We've just quantified the Euclidean distance between these trajectories and plotted it as a function from lick onset. This is just another kind of way of quantifying a little bit what I've just been talking about. So across all cell types, choice encoding increases in anticipation of the choice. As I just described, layer 5 ET cells seem to show the most pronounced encoding of choice, and this effect does not change across learning. And in contrast, layer 5 IT cells actually should show a subtle increase in distance between these trajectories from early to late in learning. So this kind of analysis effort is still somewhat ongoing, but for the moment, this kind of suggests to us that when a stimulus cannot be assigned to a category, ET neurons don't necessarily change their responses to that stimulus, whereas IT neurons may increase their choice selectivity to this uncategorizable stimuli as a way of helping to learn the category boundary. Okay, and I'll just finish with a couple of slides showing where some of this work is actually going to in my lab, because this finding of the shaping of categorical representations has really opened the door to trying to understand what is sculpting this particular response. What we essentially want to try and do is find a top-down task-dependent filter that can alter auditory cortex properties to encode task-relevant variables. This is kind of just a data dump of a whole bunch of different papers in the literature. This is a whole number of different studies that have looked at the roles of projections from higher order cortices onto a sensory cortex during a variety of different behaviors. I'm showing these distinct examples just to make a couple of key points. The first is that many different cortical areas send projections onto sensory cortices. I showed you that earlier with the rabies tracing. So if we really want to understand top-down control as a kind of a canonical circuit motif, we really need to make comparisons across different regions. All of these studies, they look at different circuits, different higher order regions, different sensory cortices and different behaviors. So if we really want to kind of understand top-down modulation, we need to compare and contrast all of these components within a unified sensory and behavioral framework. So something that we're currently implementing is a delayed version of this auditory categorization task that I've described throughout the talk. And this is so that we can start to dissociate distinct phases of the conversion of sensation into action. So in our case, these distinct phases illustrated in the top left there are stimulus categorization, conversion of this categorization into a decision and a subsequent motor plan, and then the lick, the kind of behavioral output on the right-hand side. And I think by temporally dissociating these distinct phases of the task, and then inactivating top-down inputs during these different phases, we'll be able to get a higher resolution behavioral look at how these different areas are contributing. So we're embarking, or rather Mikey Mallon, a very talented graduate student in the lab, is embarking on this wonderful set of experiments where we render the skull optically transparent and we robotically move an optic fiber to stimulate different parts the frontal regions of the brain, moving from prefrontal ROIs through motor-related ROIs to association ROIs, so that we can assay how all of these different parts of the brain interact at the level of the auditory cortex. I'm a bit of a fan of the orbital frontal cortex. On the right-hand side here, I'm just showing some kind of preliminary data showing that indeed, if we selectively express a kind of optogenetic inhibitor in the orbital frontal cells that interact with the auditory cortex, you can see that it can change the way in which the auditory cortex actually responds to these sinusoidally amplitude modulated noises. So this is super preliminary, and this is stuff that we're currently working on. So I will finish there, and I'll just acknowledge my wonderful lab, particularly Megan Arnold. She carried out a lot of the anatomical work that I've shown you. Nathan Schneider is an absolute powerhouse who carried out all of the two-photon imaging, all of the categorization work towards the end of the talk, and Becca Krall did a lot of the initial forays into getting the behavior set up and understanding how auditory cortical manipulations affect performance in that task. So I will stop there, and I'm happy to take any questions.
Great talk, Ross. We have a couple questions in the chat that I'll kick us off with. David Moore asks, all references here were to a unitary auditory cortex. But traditionally, the auditory cortex has been divided into multiple subregions. Actually, he's here, so if you want to ask the question, you go ahead.
Yeah, well, thanks. I'd just like to start off by saying a truly awesome presentation.
Thank you very much.
For somebody who's been in the field for 50 years, it's just so amazing to see how things have moved along, and congratulations. But yeah, so my question comes from that tradition, and as Kishore said, traditionally the auditory cortex has been divided into primarily tone organized versus complex sound and other stuff organized. And I just wondered, I know it's a big question, but how does your work fit into this framework?
Yeah, I mean, unfortunately, I don't have a brilliant answer for this other than to say that everything that we do, so I glossed over a lot of these details. Everything that I showed you is from primary auditory cortex, specifically A1. So we do go to great efforts to use wide field imaging as well as two-photon microscopy to map out the tonotopic structure of the auditory cortex prior to carrying out the imaging. And we specifically isolate A1 so as to not move into a lot of these different regions. From an anatomical perspective, what's really, really interesting is that a lot of these projection neurons actually do persist across different subfields in themes and variations. I can't really say too much more than that because we haven't really done a lot of that work, other than to say that people like Hiroyuki Kato is really pushing the field forward by trying to understand differential contributions of A1 versus AAF to all of these different auditory cortical subfields.
Can I just ask a quick follow-up? I mean, given that it is A1, would you predict that a lot of this stuff is going to be happening thalamically or even at lower levels?
That is a great question. That's something that we're interested with. So another area of the lab that we're wanting to, another direction that we're wanting to move into is really trying to understand how the downstream targets of these particular cell types are involved in these kind of behaviors. So does the auditory cortex send these representations to a lot of these downstream targets? With the level of A1, that's the interesting one, because A1 receives a lot of lemniscal input directly from the thalamus, whereas the layer 5 ET neurons that I showed to you, their output patterns are actually very non-lemniscal. So the output of layer 5 ET neurons projects to the non-lemniscal parts of the inferior colliculus, and they collateralize into the non-lemniscal parts of the thalamus. So it leads you into this really fascinating kind of recurrent feedback loops that are very, very kind of multisynaptic. So how those representations are propagated between all of those different synapses, I don't know.
All right. We have another question that I'll just tell you from Justin Yao. A question related to inactivating auditory cortex, that how it removes a repeated correct choice bias on more difficult trials. Does this suggest that the auditory cortex also modulates decision strategies? And could this be due to input from higher order areas like PPC, or perhaps related to sort of particular cell types as you've been playing with?
Yes, 100%. So it does change. I guess it depends what you mean by decision strategies, Justin. We've looked at choice bias, but we haven't really gone too much further than that. We could have a long conversation about this offline, about the way in which all of this stuff affects individual cell types, because we actually have a preprint on bioRxiv right now that kind of touches on this quite a lot. Essentially, it's a technically very, very hard problem, purely because a lot of these excitatory cell types interact with one another within the cortical column. So if you optogenetically manipulate one, you optogenetically manipulate everything. And we've kind of done electrophysiology to show this. If you tickle their 5ET neurons, you can see waves of activity up and down the column. So it is actually quite technically challenging to isolate individual cell type contributions to behavior. Are behavioral strategies and this kind of stuff shaped by top-down inputs? 100%. That's exactly what I was kind of trying to focus on the final slides there. I don't know what areas are going to be the most important, and that's something that we're wanting to try and find out.
Great, thanks. And we have a question from Daniel Lana, who will just ask it himself.
Hey Ross, how's it going?
Hey,
Fantastic
how's it
talk.
going? Thank you.
So those bimodal CSI distributions are just super fascinating, the fact that they evolve over time. And I'm sorry if I missed it. Have you tried silencing the ET neurons on their own and seeing how that affects perceptual categorization in the task?
Yeah. This is what I was kind of alluding to. We have this in our bioRxiv preprint, but I didn't actually talk about it. We don't see a tremendous amount. There are subtle changes across learning. Part of it is due to the way in which we were carrying out the optogenetics. The experiment that we carried out was silencing individual cell types on 20% of trials as a function of learning. And you do see a reduction in behavioral performance, but it's very modest compared to when you silence the entire auditory cortex. The biggest change that you see with layer 5 ET neurons, as well as layer 5 IT neurons as well, is that it affects learning. So it's kind of consistent with what people like Vicky Beho have shown in the past, and Hideko Inagaki from Svoboda's group has shown in the somatosensory system and motor system, that when you take ET cells out of the equation, animals can typically still kind of express the behavior, for lack of a better term, but it affects the way in which they learn. So if you silence layer 5 ET neurons, even on just 20% of trials, it affects, or at least seems to affect, learning rates, and where the animals actually plateau is a lot less. But we want to delve into that a lot more by silencing on more trials. And yeah, this is stuff I would love to talk your ear off about, because one of my postdocs is kind of looking at the differential contributions of layer 5 versus layer 6B and the cortical collicular projection, and trying to understand how those two layers contribute to this kind of stuff.
Great, thanks so much.
And then we have time for one more question from Shang, who asks, it seems in one of your slides during the training period, there's an early phase during which the population neural choice selectivity is biased to one type of category, for example, towards the slow category. Do animals in that case also show behavioral bias towards one category during early training?
Sorry, which slide did I show that?
I don't know, but is that something that you've seen? I guess, like, do you have, like, during learning data, if there's a bias towards one category neurally, is there also bias in the behavior?
Yeah, let me see if I can.
And this will be our last question.
Yeah, you can sometimes see a little bit of, part of this is just to do with the way that so yeah, sometimes there is an initial bias that is present early in learning. One thing that I want to make clear, though, is that and apologies if this wasn't clear throughout the talk, this is what I'm calling early here is early categorization. So early categorization, the animals have already learned to discriminate the two easiest stimuli. So they're already at 85% on the easy stimuli. So early in categorical learning, it's their first exposure to all of the different intermediate stimuli, but they may have already had some kind of biases as a result of the initial training. And I think that is reflected in this plot here, because you can see on the left-hand side that although the magnitude of the index definitely gets stronger, that doesn't necessarily hover around about 0 on day 1.
All right, great. It looks like we've hit 1 o'clock. Thanks to everybody for coming out for this excellent seminar by Dr. Ross Williamson. And I think this is the last one of the year. Is that right? So hopefully see you next year. Thanks, everyone.
Thanks, guys. Thank you.
And thanks, Ross.
Cheers.
Cheers.